Hi, my name is Sarah Chinn. I am a PhD candidate at University of Georgia Savannah River Ecology Lab, and today I'll be talking about the influence of intrinsic and extrinsic attributes on neonate survival of an invasive large mammal. And first, I would like to recognize my co-authors, Dr. John Kilgo and Mark Vukovich from the USDA U.S. Forest Service Southern Research Station, and my advisor at the University of Georgia's Savannah River Ecology Lab, Dr. Jim Beasley. Population dynamics, so how populations change in size and structure over time, is driven by factors such as births and deaths, environmental variation, density dependence and food availability, habitat quality, and predation. And consequently, they reflect the unique interactions between organism and its environment. And this is where individual success determines the number, spatial distribution, and genetic composition of populations. And understanding these drivers is important for determining how and why populations increase, decrease, and fluctuate spatially and temporally under changing conditions, and thus is vital to management and conservation. And for this talk, we're going to focus on the births and deaths, so the survival component. And survival is integral to population growth. It's associated with intrinsic biological attributes such as age, genetics, size, and physiological conditions, as well as extrinsic factors, things like environmental variations, so seasons, landscape characteristics, and resources. And also for species with parental care, offspring survival may be dependent on a suite of parental attributes and behaviors such as the duration of parental care, resource selection, predator defense, size, and age. Therefore, it's important to test the individual offspring, parental, and environmental attributes simultaneously when asking what's important for survival of young. Wild pigs are one of the most damaging invasive species globally. They are ecological engineers that severely impact the natural and anthropogenic environments. Thus, there's a need to know what drives population dynamics to inform population models and to effectively manage them. And most wild pig management is focused on lethal control and doesn't typically look at the biological or ecological mechanisms that affect population. And an example of this is that and it's so common that juvenile survival is often ignored in most population models. Whoop, we're going the wrong way. Okay, there we go. And this is generally because tracking adults is a lot easier. Researchers can put VHF and GPS collars onto these older age classes because they aren't growing as rapidly. And as a result, piglet survival has been widely ignored because they're typically difficult to capture, and there yet haven't been any good solutions for putting tags on them for tracking. So aside from being able to just track piglet survival, it's also important to determine what factors are affecting and influencing their survival. So few studies have actually tracked piglet survival, and none have actually successfully tracked or quantified neonate or the newborn survival. And those few studies are listed here. And most of these assess survival of larger piglets, often of unknown age, that were opportunistically captured. Two studies that have attempted to survey neonates were not successful due to poor transmitter retention or abandonment of the litter by the sow or the mother. And neonate survival studies are fairly common for other ungulates, especially white-tailed deer, which is a species that overlaps in range and habitat with wild pigs. So the objectives of our study were to quantify neonate wild pig survival, assess performance of a new VHF ear tag transmitter, and to determine which individual, maternal, and environmental factors could be important for neonate survival for wild pigs in South Carolina. We predicted that neonate survival would be greater for those that were born larger, male, and had the wild type pelage coloration. And so here I'm just showing some of the examples of the color variation we have in our wild pig population. So on the left, those two photos are the wild type, so the brown striped piglets. And there is high variability in coat coloration because wild pigs are hybrids of wild boar and domesticated pig breeds. We also predicted that neonates born to the larger size sows would have higher survival. And this is because sow size is often correlated with age and more reproductive events. These larger sows should be older and have more experience with having litters. And finally, we predicted that neonates born in the warmer months, and in South Carolina, that's March through September, would have greater survival compared to neonates born in those colder months. And that's because they are inefficient at thermoregulating. Neonates have a high surface area to volume ratio because they're small, and they lose heat very quickly to the environment. They're also born without brown adipose fat tissue and must expend energy in order to shiver to create body heat when it's cold outside. This study takes place on the Savannah River site in Aiken, South Carolina. It is a 7,800 hectare secure Department of Energy site that was established in 1952. There are about 6,500 pigs that live on site, and population is managed lethally year-round. Our habitat is dominated by longleaf pine plantations and bottomland swamps. And given the large size of our site and the lack of public access, potential predators of our neonates, so coyotes, bobcats, and possibly large raptors, are abundant and found throughout our site. We keep going backwards. We worked with U.S. Forest Service to capture females, and after they were anesthetized, we used a portable ultrasound to determine if the sow was pregnant. And if she was, we then implanted a vaginal implant transmitter, or a VIT, which is a VHF transmitter placed in the uterus of a pregnant sow that is then expelled upon parturition. When it's expelled, it keeps track of the number of half hours that it's outside the body and also sends a VHF signal to us that we can track to that tells us the birth location. After that, we put on either radio or GPS collars onto each sow to allow for more easy tracking along the landscape. And these sows with VITs were then monitored at least every other day until they gave birth. And after a sow gave birth and the VIT signaled the location and timing, we waited anywhere from about 1 to 3 days, and then we walked in on the sow, we flushed her and captured the neonates by hand. And at the nest, we photographed, counted, weighed, and determined sex of each of the individual piglets. And then we randomly chose 1 to 3 piglets of that litter to affix this VHF ear tag. And then prior to leaving, we returned the piglets to the nest and deployed several remote cameras in the area to document neonate movement, when the sow returned and when they left the nest. We monitored the neonates at least every other day until transmitter failure, which could occur by equipment failure or if it was removed by the sow, or until piglet mortality, or until 6 weeks. So we left the neonates and sows alone at the nest post-tagging for 2 days to allow for a reunion and bonding, and then we attempted to monitor them. And if we heard a mortality signal from a VHF tag, we then tracked to the tag and attempted to determine any cause of mortality if a carcass was present. And we were able to successfully track untagged neonates as well because we took and documented photos of their unique color patterns upon capture at the nest. And so all the neonates from a litter do stay together and stay close to the sow and cannot survive if they're not with her. And so we're able to sort of assume that if we get close enough to the sow, we should be able to hear her piglets with the tags. And because we use these remote cameras, we were able to determine if a VHF tag was removed or fell out versus a mortality event because we could reliably and consistently get images of sows and neonates on camera. And so we developed this VHF ear tag transmitter with Holohill. And so this is the smallest ear tag to date that's used on neonate wild pigs. It has a battery life of about 3 months and is programmed with a mortality sensor that activates after about 12 hours of inactivity. These tags are small enough and durable against removal so that we could assess neonate survival, so true neonate survival when they're just a few days old, to about 6 weeks old, filling a gap in known fate survival data for this species. And so to evaluate factors that we thought could influence neonate survival, we constructed a suite of known fate models in a Bayesian framework that included the intrinsic factors of neonate mass, sex, pelage color, and extrinsic factors such as the sow mass or sow size and season. And then we used WAIC model selection to rank our candidate models. And from this, we modeled daily survival for each neonate and grouped them by sow. And this is because we had multiple neonates for each sow, so we were able to monitor several piglets from each litter. We captured and vided 23 sows from 2017 to 2020. And for a variety of reasons, some of our vits failed. So sometimes we couldn't find the nest, or we found the nest and the sow didn't seem to have piglets. And we also had a few abandonments. So in the end, we have survival data from 15 litters that had 67 neonates, and we tagged 26 of them. And of these 67, we were able to track 50 of them until mortality or 6 weeks old. And this includes the untagged and tagged neonates from 13 litters. And then of those, 22 neonates survived to 6 weeks, giving us an overall survival rate of 44%. So here we calculated the daily neonate survival probability. And here again, our neonates are clustered by sow. So on the x-axis, we have 13 different litters. And note that P331 is represented twice. She had 2 litters in our study. And then the daily survival probability is on the x-axis. And we have our 95% confidence intervals. And our survival probabilities range from about 91% to over 99%. We also calculated the cumulative survival probability from birth until 6 weeks old for each sow. So on the x-axis, we have the number of days from 0 to 42 or 6 weeks, and then the cumulative survival probability on the x-axis. And all the way on the far right are the different sows. So each different colored line represents neonate survival probability for each sow. And so here, our cumulative survival probabilities range from about 17% to just over 82% at 6 weeks. And let's see if I can do this. You can sort of tell by this graph. We also just asked, what are the highest mortality rates for, is there a time period where we had highest mortality rates for our neonates? And that occurs within the first 10 days here. So neonates have highest hazard rates from 0 to 10 days. So when we look at our model, we found that neonates born to smaller mothers during the warm season, neonates with the wild pelage coloration, and those that were born male had greater neonate survival. And our data suggest that survival of neonate wild pigs is relatively high at 44%. And this underscores the potential for this species to expand in population size and distribution upon establishment in novel environments. Ungulate survival neonate survival for other ungulate species has a wide range from about 1 to 85% for different species. And so wild pigs kind of fall right in the middle. And we had some variation in our neonate survival probabilities between those 13 different sows or 13 different litters, but the confidence intervals all overlapped. So there doesn't seem like a significant individual difference that could affect population abundance. And from our known fate model, we found that there was lower neonate survival with larger sows. And from other studies, we know that larger sows are usually associated with larger litter sizes. So it could be harder for these sows to keep track or defend more neonates, which could account for lower survival rates. And smaller sows are usually younger and have smaller litters. So maybe they could possibly be more vigilant because they're inexperienced. And maybe it could be more manageable for them to care for fewer neonates and keep them alive. Inexperienced mothers in other species have been observed to be more anxious and attentive as well. And so looking at some of our intrinsic factors, we have one environmental factor, the cold and warm season. And again, thermoregulation might be easier during the warmer months compared to keeping up an elevated body temperature in those colder months and having to expend energy, again, because our neonates are born without brown fat and have a high surface area to volume ratio. And then looking at some of these intrinsic variables, neonates born with the wild pelage coloration had higher survival, likely because they were better camouflaged from predators. And our male neonates also had higher survival. And on average, through different wild pig populations and as well as the wild pigs born on the SRS, males are born on average larger compared to females, which could help with thermoregulation. Slightly larger body size can help with some of that heat inertia. And larger size could also give them a boost in sibling competition, possibly for these higher quality teats for nursing. And thus maybe males are able to get more of the necessary nutrients and immune antibodies that are important for survival. Our study is the first to monitor real-time neonate survival successfully, and determining survival of piglets is important for elucidating factors and drivers of population dynamics, and for developing realistic population models across their invasive range. And it's likely, and because juvenile survival is a significant factor for other ungulate species, it very well likely is for wild pigs. So it's important that we now have a little bit of information on juvenile survival as well to incorporate in these population models. And our data will inform population models for the development of effective and adaptive management strategies to ultimately reduce negative impacts of this destructive invasive species on native ecosystems, livestock, as well as human health. And I would like to thank the funders for supporting our study, including the Department of Energy, U.S. Forest Service, and the many people that help collect data from our lab, including other researchers, graduate students, and technicians. Also like to thank the folks at U.S. Forest Service for assisting with captures of the pregnant females, as well as Heather Gaia for her help with the model framework. Thank you for listening, and I hope to hear from you if you have any questions, and my email is at the bottom of this slide. Thanks.